Human metapneumovirus, or HMPV, is an aggressive respiratory pathogen driving dangerous lower respiratory tract infections in young infants, the elderly, and the immunocompromised. Classified within the Pneumoviridae family, it is a close structural relative of RSV. Despite its prevalence, it was only identified in 2001, as it is notoriously difficult to culture in traditional laboratory settings. The virus presents a clinical contradiction. It is an acute, non-latent RNA virus that the body eventually clears. Yet severe infections are strongly associated with the later development of chronic airway hyperreactivity. To find the source of that long-term damage, we have to look at how the virion interacts with the respiratory epithelium. As an enveloped virus, HMPV is wrapped in a host-derived lipid bilayer. Its ability to breach the cell membrane depends on its surface glycoproteins, specifically the attachment, or G protein, and the fusion, or F protein. Unlike more complex pathogens, HMPV does not bypass host defenses by synthesizing large, specialized structures. Instead, it utilizes minimal, highly conserved sequences of amino acids, known as functional motifs. Through molecular mimicry, HMPV impersonates host proteins to exploit existing structural templates, turning the body's own localized signaling and transport pathways against it. The viral fusion protein is responsible for the final merger of membranes, but its initial entry strategy is built on structural deception. This model of the F protein isolates the specific sequence responsible for this deception, the RGD, or Arg-Gli-Asp motif. The RGD sequence is a classic functional motif found extensively throughout human physiology. Human extracellular matrix proteins, like fibronectin and vitronectin, use this exact motif to anchor themselves to the cell surface. Because of this sequence, both the host fibronectin and the viral F protein share a perfect binding affinity for alpha-V beta-1 integrins located on lung cells. The lung cell interprets this viral attachment as a normal, healthy interaction with the surrounding extracellular matrix. This specific integrin binding signals the host cell to actively pull the viral particle inside the membrane via endocytosis. By exploiting the structural overlap, HMPV bypasses external mechanical defenses. It effectively hijacks the cell's own transport machinery to gain entry. Once inside the cytoplasm, the viral envelope fuses and HMPV RNA replication begins. To defend itself, the host cell must detect this foreign RNA and trigger the type 1 interferon pathway to alert neighboring cells. Host sensors like RIG-I and MDA5 are designed to bind to viral RNA and initiate this internal alarm. HMPV suppresses this response through its non-structural M2-2 and P proteins. These viral proteins mimic the binding interface of natural host inhibitors. They slot directly into RIG-I and MDA5, acting as a physical cap. This blockade blinds the sensors, allowing massive viral replication to continue undetected. Deeper in the signaling cascade, the viral P protein mimics the geometric interface used by host kinases. By occupying this interface, the P protein prevents the phosphorylation of interferon regulatory factor 3, or IRF3. Without that chemical activation, IRF3 cannot enter the nucleus, halting the transcription of antiviral genes. To maximize production, the virus must also ensure the host cell does not die prematurely. HMPV temporarily activates the host's PI3K-AKT pathway, delaying apoptosis to keep the viral factory operational. While this pathway is often associated with cancer, HMPV is not an oncogenic virus. This activation is strictly temporary and ends once the local resources are exhausted and the infection resolves. As replication concludes, fully formed viral particles spread back into the extracellular space to infect the surrounding epithelium. Here, the viral attachment protein uses another motif to neutralize incoming immune cells. This protein utilizes a sequence known as the CX3C motif. The CX3C motif is a structural impersonator of human chemokines, the signaling proteins normally used to recruit immune cells to an infection. By mimicking these signals, the virus tricks immune cells expressing the CX3CR1 receptor into migrating towards the virus rather than the site of infection. The virus acts as a false homing beacon, drawing defensive cells away from where they are needed most. By corralling these cells, HMPV neutralizes the first wave of extracellular defense and spreads unchecked across the airway lining. Eventually, the localized evasion fails as the viral load reaches critical mass. the immune system finally detects the scale of the threat and releases a massive, uncoordinated flood of pro-inflammatory cytokines. This cytokine storm triggers bystander activation, where large numbers of T cells begin attacking healthy host tissue. This results in the widespread death and sloughing of epithelial cells and significant swelling or edema in the small airways. The resulting mixture of fluid and dead cells forms dense mucus plugs that block airflow, leading to clinical bronchiolitis. At this stage, the molecular damage translates into a clinical emergency, often requiring medical intervention to maintain oxygen levels. While the virus is eventually cleared, the consequences of its mimicry linger in the immune memory. The combination of bystander inflammation and structural mimicry leaves the immune system sensitized to the host's own tissues. Specifically, T cells may remain primed against the lung's native collagen and elastin fibers, mistaking them for the viral motifs they were trained to destroy. This cross-reactive sensitization provides a compelling mechanism for why a brief HMPV infection is so strongly correlated with chronic airway remodeling and the long-term development of asthma-like symptoms.